Welcome to Parasitology. Today, we are going to explore a microscopic predator that acts like a Trojan horse inside the human body, known as Entamoeba histolytica. This organism might be invisible to the naked eye, but its impact on global health is absolutely massive. By the end of this lecture, you will understand exactly how it survives, how it attacks our cells, and how we can use mathematics to track its spread. To begin our journey, let us imagine drinking a glass of cool water from a stream. Unbeknownst to you, tiny armored capsules might be floating inside. These capsules sneak past the harsh acid of your stomach completely unharmed. Once they reach the safe, nutrient-rich environment of your intestines, they break open and release active, hungry invaders. This is exactly how Entamoeba histolytica operates. It is an anaerobic parasitic protozoan that primarily infects the human gastrointestinal tract, causing a severe and sometimes fatal disease known as amoebiasis. Now that we know what this parasite does, let us look at its name, which tells you exactly how it behaves. The word Entamoeba refers to an amoeba living inside the intestines. The species name, histolytica, is even more descriptive. It literally translates to tissue destroying, where histo means tissue and lytic means destroying. This perfectly describes its nature as a destructive, tissue-eating predator that feeds on your own cells once it establishes itself inside your body. Understanding its name leads us to how it survives and attacks, which involves two distinct morphological stages. First is the infective cyst stage. The cyst is a highly resilient spherical structure protected by a tough chitin wall, allowing it to survive for months in soil or water. The second stage is the active, disease-causing trophozoite. This is the motile, feeding form. It uses finger-like projections called pseudopodia to crawl along the intestinal lining, actively engulfing red blood cells and destroying host tissues. Knowing how tough the cyst is, it is easy to see why this parasite is so dangerous globally. It spreads rapidly through the fecal-oral route, especially in regions with poor sanitation. In fact, it is the second leading parasitic cause of death worldwide, right after malaria. Because standard chlorine treatments often fail to kill the tough cysts, boiling water or using specific microfilters is absolutely required. If left untreated, the parasite can even travel from the intestines to the liver, forming fatal abscesses. This rapid spread within the body brings us to the mathematics of the infection. Once the parasite reaches the large intestine, the active trophozoites multiply through binary fission, where one cell splits into two. We model this rapid population explosion using the exponential growth formula. N sub t represents the total number of trophozoites at a specific time. N sub 0 is the initial number of trophozoites that emerged from the cysts. The base 2 represents binary fission, and the exponent n is the number of division cycles. As the number of generations increases, the parasite population grows exponentially. Let us see this formula in action with a concrete example. Imagine a person swallows a single cyst. Biologically, one mature cyst releases exactly 8 trophozoites, so our initial population is 8. If they divide once every 8 hours, how many will there be after 48 hours? First, we find the number of generations by dividing 48 by 8, giving us 6 cycles. We plug these into our formula, 8 times 2 to the power of 6. 2 to the 6th power is 64. Multiplying 8 by 64 gives us a final count of 512 trophozoites. From just one cyst, we get over 500 tissue-destroying cells in 2 days. Now that we have seen how rapidly the parasite multiplies inside a single host, let us zoom out and look at the infection from a public health perspective. When a community's water source is contaminated, perhaps after a severe monsoon floods local wells, medical professionals need to assess the scale of the outbreak. To do this, they rely on fundamental epidemiological metrics. The most important of these is the prevalence rate, which tells us exactly what fraction of a population is currently infected by the disease. To calculate this metric, we use the standard prevalence formula. This formula is quite straightforward but incredibly powerful for public health interventions. The variable p stands for the prevalence rate, expressed as a percentage. The variable c represents the total number of confirmed cases, which means the number of people actively shedding cysts. Finally, the variable t represents the total population of the area that was exposed to the risk. We divide the cases by the total population and then multiply by 100 to convert the decimal into a highly readable percentage. Let us apply this formula to a real-world scenario. Suppose a rural village has a total population of 2,500 people. A medical camp tests the villagers and finds that 175 individuals are actively infected. We want to find the prevalence rate. We place 175 in the numerator and 2,500 in the denominator. Dividing these gives us a decimal value of 0.07. Multiplying by 100 yields a final prevalence rate of 7%. In public health terms, a 7% infection rate for a severe pathogen is alarmingly high and requires immediate emergency intervention. To tie all of this together, let us examine the complete life cycle of the parasite shown in this diagram. Notice the human figure on the left. The cycle begins at the top with the ingestion of a mature cyst. Follow the arrows down into the digestive tract. In the small intestine, excistation occurs, releasing the parasites. They then travel to the large intestine, shown on the right, where trophozoite multiplication happens, causing severe ulcers. Finally, the arrows point downward to show the excretion of new cysts in feces, ready to contaminate the environment and start the cycle all over again. We have covered a lot of ground today, so let us review the key takeaways. First, Entamoeba histolytica is a dangerous, tissue-destroying parasite that perfectly adapts to its environment by hiding inside a tough cyst. Second, once inside the host, it unleashes rapidly multiplying trophozoites that grow exponentially, causing severe ulcers and potentially fatal liver abscesses. Finally, understanding both its biological life cycle and its epidemiological spread is the ultimate key to diagnosing, treating, and preventing this devastating global disease. You now have a solid foundation in understanding this critical parasite.